All right, everybody. I think we're ready to start with this awesome session. So I will co-chairing this with Clemente here. So what we're trying to achieve with this session and the talk that have been selected is to define the mechanisms for chronic inflammation in CF and potentially pinpoint new therapeutic targets, and also spark a discussion on how can we start to think about immunotherapies in CF. So we're going to have 4 confirmed talks at least. We're trying to see if the last speaker can actually upload the recording for her talks. Unfortunately, she wasn't able to be here in person. We were notified yesterday about this. So hopefully she's going to be able to do that. But if not, what we're going to do after the 4th talk, we're going to bring our speakers here and have like a small panel for open discussion on how to move forward. And with that said, I'm happy to welcome Catherine from Emory University.
Hi, good morning, everyone. I'm very excited to talk to you guys today about neutrophil immunometabolism in CF. We've got nothing to disclose. Starting right into it, well, we know that there's chronic pathogen colonization and airway obstruction leading to progressive lung disease in CF. CF lung disease also has high morbidity and mortality. And looking at the role of neutrophils, we know that neutrophils are exposed to both systemic as well as organ-specific metabolic imbalances, listed there. And neutrophils infiltrate the lung at higher rates in people with CF compared to healthy controls. But often these neutrophils are unable to clear infections. Rabin and others have been part of a long history of characterizing this phenotype, which is now classified as a GRIM phenotype. That's granule releasing, immunomodulatory, and metabolically licensed. So we use a transmigration model to further investigate the mechanisms underlying this GRIM phenotype. We take blood from either CF or healthy controls, isolate the neutrophils by negative antibody selection, and then pass them through an epithelial barrier that's grown on an Alvatex filter into LTB4 or sputum collected from people with cystic fibrosis on ETI modulator therapy. Our hypothesis here is that neutrophil central carbon metabolism is deranged upon entry into the CF lung environment, and we've got a few different methods of further investigating the mechanisms underlying metabolic dysfunction in neutrophils. The first that I'll talk about is going to be extracellular flux measurements. Based on changes in extracellular pH, as well as oxygen consumption, we can get measures of glycolysis, ATP, and mitochondrial respiration. And so first, looking at your classic mito stress test, this test is done in live cells in real time, and after additions of multiple injections, including oligomycin, the ATP synthase inhibitor, FCCP, and uncoupler, and rotenone and antimycin, which are inhibitors of complex 1 and 3 of the electron transport chain, we can get various measures of mitochondrial function. So looking first at blood neutrophils from people with CF versus healthy control, immediately we can see that people with CF have increased mitochondrial respiration, both at baseline and maximal respiration compared to people without CF. And then further, if we look at our transmigration model, well, first, just to make a quick point on this, in the blood neutrophils, this mitochondrial respiration profile increase is consistent with increased substrate availability or mitochondrial mass, both of which we are planning on looking at in the near future. And moving into our transmigration model, looking both at CF and healthy control neutrophils transmigrated into LTB4 or CFASN, we can see that the cells transmigrated into CFASN in both cases have increased respiration profiles compared to those transmigrated into LTB4. However, we didn't find significant differences. So upon combining the CF and healthy control groups together, you can more clearly see that the respiration profile in cells transmigrated into CFASN is elevated. However, this is mostly due to non-mitochondrial respiration. And so this increase in non-mitochondrial respiration is consistent with increased oxidative stress and potentially increased NADPH oxidase activity. Looking a little bit further at rates of ATP biosynthesis, we can use an ATP rate assay to look at glycolytic as well as mitochondrial and total ATP. First, looking at the blood neutrophils, we didn't find significant changes between CF and healthy controls. However, upon transmigration, we can see that cells transmigrated into the CFASN, similar to a CF airway environment, have increased rates of total ATP production. However, this is primarily driven by increases in glycolytic ATP, not mitochondrial-derived ATP. Looking further at the glycolytic shift seen in neutrophils, this has already been established before that neutrophils have increased glycolysis in CF airways. But using this glycolytic rate assay, we can see basal as well as compensatory glycolysis and post-2DG or non-glycolytic acidification. And here we see a similar thing to our mitochondrial respiration profile, where we see increased glycolysis in the CF blood neutrophils compared to healthy control. However, this is primarily driven by the post-2DG acidification. And if we look at our transmigrated cells, we see that there's an increase in basal glycolysis, compensatory glycolysis, as well as post-2DG acidification. So that post-2DG acidification or non-glycolytic acidification will come back shortly. So after our bioenergetic profiling, we wanted to further characterize what the metabolites of these pathways look like in our transmigration model. So we did 1,2-C13 glucose isotope tracing, followed by mass spec using a HELIC method and metabolite analysis. So looking at glucose, you can see our two labeled carbons in red here, the 1 and 2. And just looking from our cells that are transmigrated, we immediately see that most of the glucose in our system is from the 2-carbon glucose that we, or 13C glucose that we put into the system. So that's represented by that pink bar. So if we follow glycolysis through, we start to see some interesting trends arise. Not just that there's increased glycolytic metabolites throughout our system in the neutrophils transmigrated into CFASN, but we also see an increase in the M0 isotopolog. That's the naive glucose. That's also increasing. So that could be due to various things, such as glycogen breakdown or glutaminolysis or gluconeogenesis. We did also want to look back at that non-glycolytic acidification, which we think may be partially due to the production of CO2 from the pentose phosphate pathway. And so one of these first metabolites in the pentose phosphate pathway is 6-PG. You can see it here again with our 2 carbons labeled, and still that increase in both the M2 isotopologs as well as M0. But as pentose phosphate pathway progresses, there's a decarboxylation that occurs, and one of our isotopically labeled carbons goes away. So we see the entry now of an M1 isotopolog purple bar here. However, as we continue through, occasionally you'll see pentose phosphate pathway re-entry into glycolysis, but we don't see a significant amount of that here, as we don't see large amounts of that purple bar continuing throughout down to pyruvate and lactic acid. So far we've seen that there's increased non-mitochondrial respiration, glycolytic and total ATP, glycolysis, non-glycolytic acidification, and pentose phosphate pathway metabolites. Looking a little bit further down into the mitochondria and TCA cycle, even though we don't see increases in mitochondrial basal or maximal respiration, we thought that there still might be dysfunction in the TCA cycle. And so at first, we don't see any changes in the early TCA metabolites. However, if we continue forward, we do start to see increases in succinate, fumarate, and malate. And in the future, we're going to be looking at glutamine isotope tracing to be able to determine if glutamine is contributing toward this increase in the later TCA cycle metabolites, or if there's anaplerotic pyruvate entry and backward cycling in the TCA cycle. Another note here is this pink bar down at fumarate. There was a recent study that showed that under hypoxic conditions, that cells can use fumarate as a terminal electron acceptor. And so we will also be looking at that in the future to see if that is happening in this system. So earlier I mentioned that there was an increase in the M0 isotopologs of glycolysis. And so that glucose source is coming from somewhere. One of the options that we think it could be coming from was glycogen breakdown. So we wanted to further investigate glycogen as well as the precursor for glycogen, UDP glucose. And we expected it to go down. However, we actually see glycogen increasing in our system. And so while glucose may still be broken down from glycogen, we also see an increase in glycogen. Additionally, there's other pathways branching off of glycolysis that we are interested in looking at in the future as well as now. And some of those include conjugated bilirubin production, extracellular matrix remodeling, as well as protein modifications. So looking at UDP glucuronic acid, we see slight decreases. However, UDP GLICNAC has an increase. And so looking back, here, we may be seeing increased protein modifications or potentially decreased bilirubin or ECM remodeling, but we will let you know. So in summary, you can see our summarized figure here. All the red are upregulated or increased findings that we found, and then the blue is slightly decreased, and black is to be determined so far. So in summary, transmigration into the CF lung environment increases neutrophil glycolytic metabolism, but we look forward to looking at these other pathways to determine other mechanisms of grimming in neutrophils. And finally, targeting neutrophil metabolism could be a promising strategy, and hopefully we'll be able to improve clinical outcomes in people with CF by targeting some of these pathways. So I'd like to acknowledge our lab and the Tiravansium lab, as well as our funding sources. We do have a metabolomics and biomarkers core up there. If you're interested in talking further, I'm at poster 199 right after this. So, thank you.
Yeah. We have a lot too.
Questions, anyone?
Hey, Jay Coles, Tulane, great talk. Are these assays done with human AB serum, or do you have serum in the?
No, we don't have serum in either the transmigration or in the seahorse assays that I've shown here. We don't have serum.
So given the amount of antibody in the CF lung, and neutrophils express FC receptor, how do you, have you tried maybe adding autologous serum, or in their sputum assay, there must be IgG in the sputum, so you could inhibit maybe Syk kinase and see if the FC receptor signaling is affecting the metabolism of the cells?
That's a good point. I have not personally tried doing that. I'm not sure if others in our floor department have tried doing it. My guess is yes, they have.
In the transmigration itself, that requires energy, right? So is that more dependent on oxphos or glycolysis just to get the cell across the...
Neutrophils in general are primarily depending on glycolysis. Yeah.
Thank you.
Hi, Sebastian from Colombia. Great talk. So I have basically two questions. The first one, based on the technology that you are using to detect the carbons, can you be sure that it's carbon 1 and 2 with the approach that you're using, or you just can detect there are 2 carbons are labeled?
So I guess based on our labeling patterns and looking at the pentose phosphate pathway signaling, we do see the entry of the M1 isotopolog in the pentose phosphate pathway. So we know that loss of a carbon is due to the pentose phosphate pathway. And so, yeah, I believe we do know at least that carbon 1 is labeled because we see the loss of it there.
Okay, and the second question is, from what I recall, the green phenotype is associated with a kind of an immune suppression in where they don't attack really the pathogen. But the glycolytic pathway is associated with the oxidative burst in neutrophils. So you are telling us that these neutrophils are more metabolically active to fight the infection, but the phenotype of these green neutrophils is the opposite. So how can you explain
Right. So, I mean, this is obviously not only metabolically driven, this phenotype. We also see loss of bacterial killing, which could be due to loss of phagocytosis or bacterial recognition. But we do consistently see increase in glycolysis, and these cells are alive. And even in cells isolated from sputum, they have higher rates of glycolysis and are active.
Hi, Erica Orsini from the Cleveland Clinic. Wonderful talk. It seems like transmigration is a really important part of the phenotype you're seeing. Have you thought at all at looking at mechanosensitive ion channels and how they might be working in this model, since they've been shown to regulate immune cell metabolism?
I have not, but actually we just got a new seahorse system that we will be soon trying to look further at the epithelial cells that are grown on those Alvatex plates and start to do more assays on them. So I have not thought much about the mechanosensitive genes involved, but it is a good point that there has to be some involvement of that.
Thank you.
Yeah.
Hey guys, going forward, if you can just briefly say your name and where you're from so that we all get to know each other, that would be really helpful. Not you, Katie, everybody knows who you are.
Hi, I'm Katie Heiser from National Jewish Health.
Hi.
Really nice talk, Kat. In terms of, you have the transmigration, you have the migration to the chemokine. You're seeing these changes. How much, if you just did a exposure to, like without even the transmigration, to PAMPs and DAMPs, do you think you would see the same kind of metabolic changes? How much of this is because things in the CF sputum that you're migrating toward are inflammatory mediators?
Right. I know that with just LPS exposure, we see an increase in glycolysis. But I haven't tried any other PAMPs or DAMPs other than that one. I do think you would still see an increase due to some of those.
And you may have said this at the beginning because I came in late, but in the CFASN that you're migrating towards, how is that made? Does that have all those things in it?
Yes. So it's sputum taken, it's dissolved and then spun down, and it's the supernatant of the sputum. So it's cell-free, it's bacteria-free. Yeah, you would have some of those. And I think that Rabin likely will talk a little bit about what's actually in the sputum that may be contributing toward this phenotype right after me. Yeah.
Right, last question.
Hi, Antonio Recchiuti, University of Chieti, Italy. Congratulations, a very nice presentation. An obvious question will be, what if the neutrophils come from a patient with CFRD?
That's a good point. And actually, I haven't actually looked at the demographics of the people with CF here. I think that we do have data on who has CFRD and who does not. And as we get more and more numbers, I will be making those comparisons and looking at CFRD as a factor. Yeah.
Well, thank you.
Thank you, Catherine. I'm taking over.
What's that?
I'm taking over. Let's go. So the next speaker is not Brian, but we'll take Rabin anyway.
Yeah, no, Camila preferred Brian, but sorry, he couldn't make it, unfortunately. So I'm Rabin Churvanziam. I'm from Emory and Children's Healthcare of Atlanta and Georgia Tech, and Kat made the work much easier for me because we'll be talking about the same model to mass produce or mass manufacture cells that look like the ones that we actually see in sputum from patients with CF. And this is kind of something we came up with in my lab a few years ago. Actually, Camila was involved in that as well at the time. And then I think we finally broke the really hard nut that I've been after since my grad school days. Try to figure out what is actually driving the chronicity of this phenotype. So I'm really excited to be presenting that to you guys. And as I was saying, this is really the work of a really great grad student, then postdoc, then research scientist in my lab. And then after 8 years, he had enough of me, so he actually left. And he's much happier now. That's what he's telling me. And I said, okay, I'm sad, but you're happy, so that's the most important. And then it's also the work of a really incredible bioinformatician that is working 100% of his time, maybe even 120%, even though it's not mathematical, in our lab, and incredible scientist, Diego Moncada. These are my disclosures. And I'm going to go quick on the actual background, because I mean we all know what we're talking about. This type of picture that you see in CF, where small airways, then larger airways, and not necessarily alveoli until late in the disease, have this pattern where the lumen is obstructed by a lot of neutrophils and bacteria and extracellular polymers like mucins and DNA and actin, etc. So these are not great to allow for airflow, obviously. But this is a picture that doesn't completely represent what we have in patients with CF. If you look at the evolution of disease, it's really patchy. So you have some areas that are like this, and then you can have a septum, and the airway right next is actually free. So it's interesting that it is not something that would be expected if you had a primary deficiency in CFTR, messing with mucociliary clearance, and then leading to bacterial recruitment, then to inflammation. So I've been really kind of obsessed with this problem since I started as a grad student, and I really think at this point this is not the model. It is not obstruction, infection, and inflammation. It's tiny levels of obstruction in certain pockets of the small airways, inflammation, then recruitment and selection of particular bacteria. Okay, and that changes completely the way we should be looking at this disease and treat it. Because ultimately, if the disease is chronic, it's because you have chronic crosstalk between those different cells. The epithelial cells obviously are abnormal, but they are not wrecked. They are repaired. They're not properly functioning, but there's no translocation of bacteria. There's no sepsis in patients with CF. So we actually know now that the neutrophils and the macrophages are sending repair signals to the epithelium. So these cells are not really inflammatory as we define them, but we do have a lot of neutrophils in the lumen. We know they talk with other cells, and we know they also talk with bacteria, viruses, and fungi. The organisms that we see in the long run are actually selected and I would say commensalized. So they're not pathogens. This is not a host pathogen interaction we're looking at. We're looking at a host commensal interaction. Okay? So again, like we really have to change, I think, our views and the way we talk about this disease, because this is not what's in the textbooks. This is work we did. It's kind of a summary. We actually published with Camila quite a few years ago, actually. 10 years. I didn't know it was 10 years. Okay. We were so young at that point. Me too, I guess. So we really ended up, asking the question of how does the disease start? And to address that, we went to our colleagues in Australia, Steve Stick and Sarat Ranganathan, and they had a really thriving prospective BAL study going on from newborn time up to 9, 10 years of age. So we had the ability to look at the disease longitudinally along with CT scans and metagenomics of the microbiota, et cetera, et cetera. So we were able to really try to derive a good picture of what was happening in the babies with CF. And we found that essentially everything starts in the small airways, in pockets of the small airways, and there's some evidence of abnormal mucins, which we got with our collaborators at UNC, Chuck Esther in particular. And we find that the first thing we see is epithelial dysfunction and some macrophage exhaustion phenotype. And right around that time, neutrophils start to come in, and they acquire the phenotype that Kat was mentioning, the green phenotype. And I'm going to go a little bit more in detail about this. At that point, none of the typical opportunistic pathogens you see in CF are present. Doesn't mean that they haven't come and left. We cannot exclude hit and run, but we can exclude colonization. Then the second phase happens where neutrophils start to dominate, and this is where, after a while, you start to have visible plugging, a little bit of bronchiectasis appearing as well. In the first years, that bronchiectasis is actually sometimes reversible, but as you advance in age, it becomes irreversible, and we can see that through longitudinal CT scans. And I see I'm already taking too much time, so I'm going to go a little bit faster. This is where you start to have the selection of bacteria that are commonly seen in patients with CF, Haemophilus influenzae, Staph, Pseudomonas. And this is what we saw in the prior picture of the tissue. Generally, bacteria are quiescent. They've lost a lot of their ability to do damage, so they're really commensals. They are generally in an exopolymer. You have neutrophils stationed all around them, controlling them, but probably feeding them with certain metabolites like nucleotides and amino acids. And then you have also some macrophages around. So this is kind of the picture of chronic CF. And as we look at this, we see obviously airway obstruction increasing, oxygen levels decreasing in those areas, lactic acid levels increasing, pH decreasing, free elastase and free DNA increasing as well. Okay? All right. So what is really germane to all of this is the appearance of that phenotype that Kat talked about. And this is just a slide that summarizes all the studies we've done in vivo. That is, we've actually gone and taken the BAR from the babies or the sputum from adolescents and adults. And we found, first of all, and it's not a surprise anymore, but it was a surprise, and it cost me 3 years of my life to convince reviewers it was true, that there actually are live neutrophils in the sputum of patients with CF. People would collect the sputum and trash it, and I would cry, and I would go to the docs when I was at Stanford at the time. I was saying, please don't trash this. Give it to me. I'll find a way to separate the different components, and I'll do fancy flow cytometry to show that there are non-apoptotic live neutrophils in there. And I did it, but still it took me a little bit of time to show that these cells are actually not just live and non-apoptotic. They are actively releasing their elastase and myeloperoxidase rich granules. Okay? And we know elastase and myeloperoxidase levels are really great biomarkers, probably the best we have still of lung function and survival in patients with CF. We showed also, and that was interesting, that these cells are downregulating the activities of both T cells and macrophages. They do remotely control T cells, primarily through the secretion of that enzyme arginase 1 that depletes arginine from the milieu, and without a proper level of arginine, T cells cannot assemble or stabilize their T cell receptor. So it's a really early block to T cell function. T cells cannot recover from it. There's no way, okay? But they also block macrophages, this time through checkpoint inhibition, direct contact through PD-1, PD-L1 signaling. So actually a paper by Camila, this one. See, I'm giving to Caesar, what Caesar deserves. But most interestingly, I think what we found, and again, we were pushed by reviewers, so I mean, I have to give them credit. They give me, difficulties throughout my life, but oftentimes they ask the right questions. Okay, these cells are alive and non-apoptotic. Are they about to die? Okay, how do you determine that for a neutrophil that anyway will last for just 2-3 days maximum? So we went in and we did phospho signaling of cells out of the sputum, and we found that they activated the TOR pathway, which is really the core anabolic pathway that you see in cells in cancer, etc. And then we did some studies with metabolic SRC transporter probes. We found increased GLUT1. We found increased branched-chain amino acid transporters, inorganic phosphate transporters, etc., etc. And then again, a study by Camila just before she left the lab, she showed that the cells in the airway actually have increased RNA, and that's essentially going along with this phenotype. What she showed also in this paper is that if you block de novo transcription by these cells, you recover bacterial killing, meaning the reason why bacteria are not killed in these patients' lungs is not because you have resistant bacteria from the beginning, is because the immune cells are deciding not to completely clear. They're allowing for a certain amount of bacteria to stay in the lung. That's critical, because if we want to clear those infections, we have to take care of the decision by the host to not kill those bacteria, right? Okay, so I'm going to go a little bit faster now. These cells, the green ones, we think they are a fate that's completely different from phagocytic and necrotic neutrophils. Why? Because this depends on where the primary granules, the ones that contain elastase and myeloperoxidase, go. If they go to the phagosome, you have a phagocytic neutrophil. If you go to the nucleus, you have a lethal netosis phenotype. If the granules go to the membrane and the contents are exocytosed, you have a GRIM. These are all the papers we've published describing this phenotype. We've done some in CF, obviously, but we've done quite a lot in COPD, neutrophilic asthma, pediatric ALI, ARDS, including in the context of severe COVID. We find these cells in all these diseases. And we think that if we figure out a little bit better what they do, we can come up with a way to manipulate this fate and improve all of these diseases, right? Okay, biomimetic model we developed in the lab. Kat talked about it already. With this model, we can take the cells after putting them into a chemotractant control or CF airway fluid. I'm going to tell you a little bit more about this. This is the big question. What's in there that's promoting the phenotype? And I'm going to give you the answer this time. Okay, we've been discussing that for years, but we have the answer. So in that particular study, doesn't matter what the status of the neutrophil or the epithelium is. It's just the exposure and the transmigration, the combination of the two. You can basically recapitulate the exocytosis of the elastase. So this is done by flow cytometry, and you can recapitulate the lack of bacterial killing. That gives you a lot of leeway to test drugs. We found also, not shown here, that these cells are anabolic. They're highly pinocytic. They're transcriptionally reprogrammed. And we know now that they recruit each other in a feed-forward manner in a transmigration model, meaning you take these cells out here, even if they start from a normal individual, you take them here, you make them make their own medium, you put the medium back here, and you get another wave of cells that look exactly like that. Okay, what's in the ASN? The ASN is full of cells, bacteria, extracellular vesicles, metabolites, PAMP, stamps, proteins, lipids, like you name it. So we can do non-chemical dissociation of this at 4 degrees, so we don't activate the cells. Differential centrifugations, and we can separate different fractions. We can get in particular extracellular vesicles. Okay, extracellular vesicles, we call them Gen 1 because they're just coming from the sputum. We can also make them from cells that have been reprogrammed in the model. They're Gen 2 and Gen 3. We can do recursive programming like this. What's important about this is we have extracellular vesicles that are coming from bacteria, from epithelial cells, from macrophages, from neutrophils. Fortunately, when cells make extracellular vesicles, those vesicles bear through their surface transmembrane proteins that are coming from the cells of origin. So you can tell based on those which cells have made those different EVs, and you can even deplete the fraction of those using antibodies. Those EVs are present in staggeringly high numbers in sputum from patients with CF. You're looking at 10 to the 11 to 10 to the 12 EVs per ml. So that's about easily 100 to 1,000 EVs per cell. that you get from the same sample, okay? So it's a lot. We did in our transmigration the following experiments where we did the cells in LTB4, they don't exocytose much, CFASN, they exocytose a lot. You remove the EVs, they stop degranulating. You add back the EVs, they degranulate. You remove specifically the neutrophilic EVs, they don't degranulate. There's not an effect due to the either epithelial or macrophage EVs. Bacterial killing is exactly the same. They lose the bacterial killing only when the neutrophilic EVs are present. What's in those neutrophilic EVs? We can do transcriptomics, we can do a lot of different omics. I'm not going to show you all the data, but I'm going to show you the transcriptomic data. We find that the EVs made by blood neutrophils compared to LTB4 recruited neutrophils in blue compared to CFASN recruited neutrophils are completely different in terms of transcriptomics. This is a principal component analysis. And the most differentially expressed RNA is a long non-coding RNA called metastasis-associated lung adenocarcinoma transcript 1, or MALAT1, which is, you recognize that by its name, has been shown in the context of lung adenocarcinoma metastases, which happen to be really rich in neutrophils. And now that we're doing these studies with our colleagues in oncology, we find that they have green neutrophils in the brain metastases. MALAT1 is low in the cells, but really high in the EVs they make. So they make a lot of it, and they ship it to the EVs, and then they send the EVs out. The EVs now go back to patients. We measure MALAT1 in the EVs in the sputum, and that correlates really nicely with FEV1. You also see a decrease in the MALAT1 content of the EVs as you go away from an exacerbation episode. So that's potentially a really nice biomarker. What's in the cells that's responding to MALAT1? We looked for transcriptional regulators in these cells, same method. We find HDAC11, histone deacetylase 11, class 4 HDAC, alone in its class. It's mostly a D-fatty acylase, unlike its name says. So it's very active in post-translational modification of proteins. It is upregulated over time in the model, pretty highly. So we can stop the transmigration at given times and look at the kind of dynamics of different genes, and we see that highly upregulated. This is in log2 fold change. Fortunately, there's a bunch of inhibitors. Some of them not so specific, but this one really specific, C17. So we went ahead, used C17 in our model. What do we see? That it doesn't change degranulation when given to normal neutrophils, but it reduces degranulation when given to the CF conditioned neutrophils. And it does allow for an uptick in bacterial killing. We can do the same thing. Instead of doing neutrophils, we can do monocytes, we recruit them and then differentiate them, and then we can look at whether they kill bacteria as well. This is work by a postdoc, Deepali Luthra, it's poster 177. She found that at high MOI of 10, the macrophages that are recruited through the same pathway into the CF-ASN lose their ability to kill bacteria and regain it when exposed to C17. So we think the macrophages are also tolerized towards bacteria through these EVs. Are MALAT1 and HDAC11 the same pathway? Yes, because what we can do is we can transfect blood neutrophils with plasmids, and we can make them express either HDAC11 or MALAT1, and we can treat them with the inhibitor. And you can see that, for example, if you force MALAT1 into a normal neutrophil, they stop the killing, and you can inhibit this effect by C17. You can do the opposite by putting an siRNA against HDAC11 or siRNA against MALAT1 in the CF neutrophils. Same thing goes for exocytosis. The exocytosis is also dependent upon MALAT1 and HDAC11 in the normal neutrophils and in the CF neutrophils. Same idea, we transfect with RNA. Okay, how about animals? Well, MALAT1 is not well conserved in rats or in mice, but it's in ferrets. At least the sequence that we saw that was highly upregulated in CFASN is not present in these small rodents, but it's present in ferrets. So we talked to John Engelhardt. We had some early studies with him showing that MALAT1 is highly upregulated in G551D BAL. He pointed us to Southern Research, who worked with Shahab and Alison, Lauren, and Doug over there. We got access to G551D ferrets. We treated them with C17 in vivo, just short-term experiment. We found after 2 doses, 6 days, delivered intratracheally, that interferon alpha was going down, IL-8 was going down. This is a marker of elastase activity going down. And most importantly, our MALAT1 in the EVs going down pretty dramatically, about 16 to 32-fold within 1 week. So that's pretty encouraging. Okay, so I'm going to conclude. I'm already like way above time. Sorry for that, guys. So this fate of neutrophils we discovered is causing active bacterial tolerance. And it's caused by the presence of MALAT1-rich EVs as extracellular vectors that reprogram the cells. And since we now know what to look for, we find it in the other diseases where we found those cells. This is not CF specific, but CF is pushing this fate of neutrophils in the lungs specifically, in those pockets where you have mucus aggregation and stasis. And we have HDAC11 as the intracellular regulator of that fate. And siRNA therapy can inhibit programming, restore bacterial tolerance in vitro. The drug, the small molecule HDAC inhibitor, can counter GRIM programming both in vitro and in ferrets. We have uncovered, we think now, finally, the loop that's responsible for host-directed tolerance of bacteria. And we need to go after this, and we think we can have that way a mutation-agnostic therapy that can actually synergize with modulators and antibiotics. Most importantly, we have actually a companion diagnostic and a way to look at the effect of the drugs because the MALAT1 in the EVs is resistant to freeze-thaw, has a high dynamic range, is quantitative, it's sensitive. So we're proposing actually this round to the CF Foundation to give us money to explore that as a biomarker for gene therapy and a bunch of other indications in CF. So I'm going to leave you with this updated paradigm. The cells come, they're naive, they get into the lung, they're exposed to the EVs left by the other cells, and they acquire this phenotype, and it's basically a cycle. When the cells are in this state, they induce bacterial tolerance also by the MACs. If we treat with the HDAC inhibitor, we get into the healthy cycle, where the neutrophils don't exocytose, they clear the bacteria, they don't make a lot of EVs, there's no MALAT1 in the EVs, and they can really work well with the MACs and clear the bacteria. These are the people who are involved. The lab has a lot of people, so pretty much everybody lend a hand, especially the people who are underlined. I really want to acknowledge Brian and Diego again, and funding by the foundation, by NIH, Endowed Chair, and then the Jora Research Alliance also for some of the HDAC11 studies. And I'm going to leave it at that, and I'm going to take questions. Thank you.
So,
Raveen, you've taught us so much over the years. You get all the time you need, man. We're all very thankful.
Thank you. I appreciate it.
We got time for maybe 3, 4-ish really quick questions.
Kong Chen from University of Pittsburgh. Real quick, a great talk first. Are epithelial cells receivers of those EVs?
Yes. They react, and part of what we found is also going to be in this paper we have in close to submission now. We find a lot of pro-repair and pro-antioxidant response signals that are sent by the neutrophils to the epithelial cells. So the epithelial cells become more resilient when they receive the EVs from the green neutrophils.
Sorry. Hi, Sebastian, Columbia University. So this is more kind of a philosophical question, okay? So I would like to have your take on this. So are these green neutrophils part of the problem or not? Why I'm saying this is because if we consider evolution, we have evolved tolerance to protect our organs from immunopathology by attacking organisms. For example, the microbiome. If we don't tolerate the microbiome, we will die of the immunopathology. And you're saying that these green neutrophils have a tolerogenic phenotype. So is this a mechanism that the host is using to try to protect the lung to not induce any immunopathology by preventing their attack to the
absolutely.
Or they are part of the problem?
Absolutely. I mean, absolutely. It's part of the solution short term, because you're not producing like a full-blown, lung injury. And same in the metastatic tumors, we find them as well, and it actually tolerate the system to the tumors in that case, which is really interesting.
Tumors, they exploit the tolerance.
Exactly. So tumors, a lot of tumors exploit the GRIM phenotype to actually block T cells and allow for angiogenesis, and those cells are actually highly metabolic, as Kat said. They are. I mean, I don't know if you remember, part one of like Back to the Future, when at the end, like Doc comes back with a flying car, and then he takes the trash and puts it in the flying car and says, it can run on anything. I know in 2020 at the time, but they didn't know what the future held. These cells run on anything, and then they basically take everything that they have around. They also promote proteolysis, so they take some of the stuff that they degrade and put that into anaerobic metabolism, put out nucleotides, put out amino acids, and then everybody around them is using that to tolerate the new state, because it's a hypermetabolic state to maintain and aggregate with a lot of bacteria, a lot of immune cells, and so on. It's not great for the body, but it prevents damage. In the long run, you basically suffer from this. But in the lifespan of a neutrophil, which is just 2 days, they might not see a change, or lifespan of a given bacterial cycle, they might not see the change either. So for these short-lived cells, the bacteria and the neutrophils, it's all cool.
Thank you.
Yeah.
Great talk. This is Chris from Cincinnati. So my question about the HDAC inhibitors has been bothering me for a while, because it seems like there could be a lot of off-target if it's given systemically. And obviously in cancer, you're sort of in a last-ditch effort. So if you would envision someone getting these long-term, do you have any idea about off-target, and have you seen anything in the ferrets?
It's a great question. So in the ferret, we don't know yet, because we've just done acute sort of dosing, but we didn't see anything, I think in 10 days or so, right, Shahab? Yeah, in 10 days or so, we didn't see anything. HDAC11 is misnamed, okay? It's mostly a D-fatty acylase. It's a class 4 HDAC. It's the only one in its class, has a very different mode of action, and it's not expressed in all cells, unlike the other HDACs. It's expressed mostly in epithelial cells, in neutrophils and macrophages, and a little bit in muscle. And now people find them in metastatic tumors as well. We don't know whether it's the tumor itself or whether it's the associated neutrophils. But essentially, we think HDAC11 is a pro-tolerant HDAC. So it's part of the same pathway, but we cannot exclude off-target effects, obviously. So part of what we're doing is working with the Shinazi group, which is really, renowned in drug development, to try to get, a little bit of medicinal chemistry, a little bit of, magic from these guys, medicinal chemists, to make the next generation of inhibitors.
Cool.
Thank you.
You want to go ahead?
All right.
Thank you, Ravi.
Thank you, Ravi. All right, so moving on with this tour de force, we have Mahesh Shesta. Let me, coming up. Let's put up his talk up there. And he's going to talk to us about the LC3-associated phagocytosis and cystic fibrosis. Welcome.
Thank you. Good morning, everyone, and thank you for this opportunity, CFF. These are my disclosures. Despite significant improvement in cystic fibrosis therapy with the advent of CFTR modulators, there is persistent infection and inflammation in people with CF due to the impairment of phagocytosis of CF pathogen despite CFTR modulations. Hence, there is urgent need to investigate the role of innate immune dysfunction and on studies on host-directed therapies. LC3-associated phagocytosis LAP is an emerging non-canonical form of autophagy, which is selective for pathogens. LAP has been implicated in clearance of several CF-related pathogens like Pseudomonas and Burkholderia. However, the fundamental aspects of LAP are altered in CF, including decreased NOx assembly, reduced LC3 decoration of phagosomes, and decreased phagocytosis of CF pathogens, which we have reported in the past. Here I briefly take you to the world of LAP. LAP is initiated when a phagocyte, like macrophages, encounters a pathogen in its extracellular environment. This leads to a series of molecular reactions leading to the engagement of cell surface receptors like TLR to the pathogen. This leads to the internalization of the pathogen inside the phagosomal membrane. At this point, LAP has been also reported to sequester phosphatidylserine in the phagosomal membranes. We'll come back to this story at the later point of this presentation. So internalization of the pathogen leads to the formation of PI3K class 3 complex on the phagosomal membrane, which is stabilized by the binding of Rubicon to the Beclin 1. Rubicon binds to the Beclin 1 and phosphorylates VPS 34 to shut down autophagy and kick off LAP. So Rubicon plays a critical role in the initiation of LAP. PI3 complex then produces phosphoinositol 3-phosphate on the phagosomal membrane, which acts as a docking site for the NOx2 complex proteins, which releases reactive oxygen species in the phagosomal lumen. Further, Rubicon binds to the NOx2 complex and stabilizes it. This stabilization of NOx2 complex leads to the conversion of cytoplasmic soluble LC3-1 into lipidated membrane, phagosomal membrane bound LC3-2 and marks it as a liposome. This liposome then binds to the fuses with the LAMP1 positive lysosome to form a complete liposome. Inside the liposome, the pathogen is neutralized by various reactive oxygen species like superoxide, hydrogen peroxide, and hypochloric acid, and then followed by the lysosomal degradation to the subcellular building blocks of the cells and recycled back to the cell. Here I quickly outlined the difference in autophagy and LAP. Although they look similar on the surface, and even though LAP utilizes some of the autophagy machinery like ATG proteins, they are initiated through different mechanisms. While autophagy is initiated by the starvation or through the accumulation of dead and damaged organelles, LAP is initiated by the presence of phagocytic cargo in its extracellular environment. LC3 decorates double membrane autophagosomes, whereas in liposome, LC3 decorates a single membrane vesicle. Both autophagosome and liposome binds to lysosomes, which are LAMP1 positive, but autophagy is mainly involved in cellular homeostasis, whereas LAP acts as an innate immune response to the pathogens. This electron microscopy image shows acts as a premise for our current work, where in our publication related to autophagy work in CF, we showed that when CF MDMs were treated with interferon gamma, there was increased production of autophagy, autophagosomes, double membrane autophagosomes, whereas in the CF MDMs untreated with interferon gamma, we saw more liposome formations, which are single-walled. So CF pathogens are readily cleared by autophagy in non-CF macrophages, but autophagy is defective in CF. And there is a broad off-target effect to target autophagy. So we ask the question, can lab compensate for autophagy? As a previous for our current work, we also reported recently that ENaC is hyperactive in CF, which targets various lab components like NOX2 complex and leads to the macrophage dysfunction. So this leads to our hypothesis. CFTR ENaC axis leads to the dysfunction of lab, causing increased inflammation and infection in people with CF. We use monocyte-derived macrophages, MDMs, and alveolar-like macrophage-like cells as our models to prove our hypothesis. Here I briefly describe the schema how we generate these cells in the lab. Monocyte-derived macrophages, MDMs, are macrophages differentiated at the site of infection in lung from systemic circulation of monocytes, whereas alveolar macrophages are lung-resident cells which act as a first line of defense against infection in lung. Here we show the cell surface structure and morphology of MDM and AMLs. While MDMs are irregular shaped and rough in cytoplasmic appearance, AML seems more round and less rough, and they both resemble the lung phenotype of these cells. So Rubicon is a critical regulator of lab, and Western blot analysis of CF MDMs infected with dominant CF pathogen Burkholderia showed decrease in Rubicon expression, which was very distinct during the treatment with NOX agonist PMA and also during autophagy inhibition during infection. Similarly, we found decreased Rubicon expression in both Pseudomonas treated group in CF and non-CF MDMs. However, we found that CF MDMs do not mount similar response to PMA during autophagy inhibition during Pseudomonas infection. We also found decreased colocalization of Rubicon with zymosin particles, a lab stimulus, and LC3 in CF MDMs as compared to non-CF MDMs showed in these white spots, that is yellow arrow. We looked at the functional aspects of lab and found the decreased liposome formations in CF MDMs treated with zymosin in presence of autophagy inhibition as compared to non-CF MDMs measured by confocal microscopy of LC3 associated liposomes. We also found decreased phagocytosis of zymosin particles by CF MDMs as compared to non-CF MDMs as measured by flow cytometry detection of fluorescently labeled zymosin particles. We also found decreased ROS production by CF MDMs upon zymosin stimulation as measured by DCF assay compared to non-CF MDMs. To emphasize on the role of ENaC in macrophage dysfunction and the lab dysfunction as well, we treated CF MDMs with broad ENaC inhibitor amiloride and specific ENaC inhibitor SPX1 in presence of zymosin stimulation and found increased expression of Rubicon in both inhibitor treated groups. We also found similar results with AML cells. Now coming back to the previous story of sequestering of phosphatidylserine by lab receptors. Recently it was published that lab receptors sequesters phosphatidylserine in phagosomal membranes. In baviteximab seems to target phosphatidylserine in tumor and infected cells, but spares normal cell. Baviteximab is a investigational chimeric mouse human monoclonal antibody, which is known to promote M1 differentiation in macrophages, thereby increasing its bacterial killing through production of ROS and nitric oxide. Initially, we did the test study to see cellular toxicity of the host cells of baviteximab and found dose-dependent increase in cellular toxicity above 10 nanogram per mole. So we decided to go with this concentration for our test. So we treated CF MDMs with baviteximab during infection with Pseudomonas, and also in isolation or in combination with rapamycin, mTOR C1 inhibitor. And we found increase in bacterial killing in baviteximab treated group during infection with Pseudomonas, which was not enhanced by the addition of rapamycin in the treatment group. In summary, Rubicon is critical regulator of LAP, and we showed that it's reduced in CF. We also showed that inhibition of ENaC potentiates LAP response, and we present baviteximab as a novel therapeutic candidate. For future directions, we would like to set up co-culture methods using different CF pathogens like Pseudomonas and staph, and also would like to work on further future mechanistic studies of LAP. We would also like to look at the impact of CFTR and ENaC related defects on LAP and pathogen clearance using translational mouse models. With that, I would like to thank my current and previous lab members and human specimen course for providing precious samples and funding support from CFF and NIH. Thank you.
Any questions?
I can start while people get there. So thank you so much. That was really interesting, Mahesh. And you know, I'm not ashamed to say that it's the first time that I hear about this LAP mechanism. So, I have a couple of questions. I have many questions, but the two questions here are in terms of the overall bacterial killing that happens when in a CF airway. What percentage of it, how much of it would occur through this versus other canonical mechanisms of bacterial clearance? And then the other question I have is, do you have a sense of how treatment with ETI, for example, of treatment with modulators would affect this process? Because it seems that there's a difference in the prevalence of Pseudomonas to some degree after people are treated.
Yeah, thank you. Thank you for the question. Yeah, in terms of the first question, so I'm not 100% sure how the difference in lab, I don't know. I mean, it would be difficult to look at how lab versus autophagy is, you know, controlling the bacterial killing. So, but we do show that like by most of the experiments we do, we inhibit autophagy so that we are just looking at the lab killing. Does that answer your question?
Well, I mean, other mechanisms for bacterial killing aside of regular LAP. So like regular, you know, phagocytosis and other mechanisms of bacterial clearance that are the standard beyond I mean, not the more canonical mechanisms, not necessarily autophagy, which is not really per se a bacterial microbial clearance mechanism.
Yeah. That's why we look at lab, because we cannot target autophagy due to its broad off-target effects, because mTORC1 is involved in the cellular proliferation and metabolism, right? So that's why we want to promote lab as a therapeutic, as potential to promote bacterial cleaning.
Great, thank you.
Antonio.
Can I have a hi? Very interesting. Two questions. One, if you think that this mechanism is also relevant to neutrophils, and the second one, if in macrophages this mechanism will be also important for the antigen presentation process.
Yeah, we haven't looked at neutrophils, but we think it should be relevant in neutrophils as well. What was the second question? Sorry.
The second question was if this mechanism can be also important for the mechanism by which macrophage make up the antigen presentation to the other immune cells, to the T cells, for the antigen processing.
Yeah, we haven't looked at that, but most likely it would be relevant.
Okay, thank you.
Thank you.
Thank you.
Thank you.
All right. The next talk will be from Ravi. We're going to hear about macrophages a little bit more.
Good morning, all. I'd like to thank the chairs and NACFC for giving me this opportunity today. I do not have any disclosures related to this presentation. In this talk, I will be... Oh, sorry. In today's talk, I will be discussing about our ongoing work where we are investigating the fate of alveolar macrophage in CF during inflammation, the mechanisms associated with it, and a potential therapeutic strategy. It is very well known that in CF, macrophages are dysfunctional and they contribute towards hyperinflammation by releasing excessive pro-inflammatory cytokines, and also they contribute towards defective host defense by due to their poor bacterial killing abilities. In this study, I will be mostly focusing on alveolar macrophage dysfunctionality in CF and the mechanisms associated with it. Again, it is very well known that alveolar macrophages are the primary tissue resident macrophages which are crucial in maintaining the lung homeostasis and host defense. And it is very well documented that this alveolar macrophage dysfunction happens at early stage in CF children. So it has been reported their numbers are abnormal, and these alveolar macrophages express less scavenger receptors, making them poor at bacterial killing abilities, which makes it important to understand the dysfunctionalities in this alveolar macrophages. In non-CF lung, during bacterial infection, alveolar macrophage acts as a frontline soldiers in killing them. And during this killing process, some of the alveolar macrophage undergoes programmed cell death. And the lost alveolar macrophages are replenished by their self-renewable mechanism or by monocyte differentiation. Whereas in CF, we hypothesize that the programmed cell death happens at much higher extent, and also they fail to replenish the lost alveolar macrophage due to impairment in the proliferation abilities, and also they possess abnormal monocyte differentiation, which will again contribute to the hyperinflammatory environment. So the loss of this alveolar macrophage has been well documented by Dr. Brito and his lab using the adult sputum samples via single-cell RNA-seq, where they were able to identify the tissue-resident alveolar macrophage populations along with the other myeloid and lymphoid cells. What was interesting was when they compared the CF samples with healthy donors, they saw the less number of tissue-resident alveolar macrophages. So, which made us curious what is happening to these alveolar macrophage. To study this in laboratory setting, we developed a mouse model where we take wild type and CF knockout mice and nebulize with LPS 3 times consecutively. So we give 1 dose a day. Post 24 hours 24 hours of last LPS, we isolate bulb cells and lung cells and do the flow assessment. When we check the wild type mice post 3-time LPS, we saw the presence of tissue resident alveolar macrophage expressing cyclic F positive and CD11C positive markers. At the same time, we also saw the presence of monocyte derived macrophages. But when we looked at the CF mice, we saw the similar reduction of alveolar macrophages post LPS, which was quite significant as well. Not only in the CF model, we also saw the similar reduced number of alveolar macrophage when we performed Pseudomonas aeruginosa infection in these mice. So we were able to establish the models where we can use them to study what is happening to these alveolar macrophages. But before moving to study what is happening in vivo setting, we took a step backward. So we wanted to assess what are the mechanisms associated in these dysfunctionalities. So to do that, we isolated primary alveolar macrophages from wild type and CF bulb and exposed them to LPS. Upon LPS exposure in wild type alveolar macrophage, we saw the cell spreading and formation of lamellopodia and phellopodia. Whereas when we looked at the CF knockout alveolar macrophage, this was completely dysregulated, and we also saw the defect in the cortical actin arrangement. And at the same time, they failed to spread. So due to this defective actin cortical organization, we also saw the increased release of LDH into the supernatant, which suggests that these cells are more prone to undergo cell death. So since we see the cytoskeleton dysregulation, we asked what are the proteins that are involved in maintaining the cytoskeleton arrangement and are they dysregulated? And one such actin binding protein that we have been extensively studying in our lab is ezrin. As I said, ezrin is an actin binding protein. Upon activation, it cross-links between plasma membrane and the actin cytoskeleton, which will make the facilitated arrangement of the cell morphology and also helps in facilitated signal transduction pathways such as cell survival pathway via PI3K AKT and also cell morphology and cell adhesion pathways. In this talk, I will be focusing on cell survival pathway. So when we looked at what is happening, how the expression and localization is affected in wild type and CF alveolar macrophage in response to LPS, we found that in wild type alveolar macrophage, there is a nice colocalization of ezrin in the cytoskeleton region. However, that was completely impaired in CF knockout alveolar macrophages. Not only the localization, but also the phosphorylated levels of ezrin was significantly downregulated, and its downstream phospho-AKT levels were also downregulated, which suggests that these CF alveolar macrophages might be more prone to undergo cell death in response to LPS. So then we were curious to study, are these defects ezrin dependent? So to study if they are ezrin dependent or not, we developed a mouse model where we specifically knocked out ezrin in myeloid cells. By extracting the monocytes and macrophages and exposing to LPS on collagen surface, we saw the similar dysregulation or defect in the actin organization in loss of ezrin. And we also isolated the alveolar macrophage from these mice and then exposed to LPS. We saw the similar downregulation in the phosphorylation of AKT. At the same time, we also saw these cells are less viable due to loss of ezrin when they were exposed to LPS. Not only the cell survival abilities, but we also checked the functional aspects like phagocytosis. We saw that loss of ezrin made these alveolar macrophages to be less phagocytosis in response to LPS. So knowing the importance of this ezrin in altering the cytoskeleton rearrangement, we then asked what is happening to this ezrin, what causes its reduction. So we first assessed the transcriptional levels in the wild type and CF knockout alveolar macrophage. In response to LPS, we saw the increased expression of ezrin in both the phenotypes. However, we did not see any difference in the mRNA levels in response to LPS. So which made us think that this could be degraded at protein level. So during our literature survey, we found that the phosphorylated ezrin gets degraded by a calcium-dependent cysteine protease called calpain. So as a therapeutic strategy or to restore the phosphorylated ezrin, we used a pharmacological inhibitor, calpeptin, to inhibit this calpain and to see if this ezrin gets restored. So interestingly, we saw the restoration of phosphorylated ezrin in CF alveolar macrophages when they are exposed to LPS, and they were comparable to wild type. And we also saw the restoration of phosphorylated AKT levels, which is downstream the ezrin pathway. And this also highlighted the importance that maybe inhibition of calpain might help in the cell survival aspect as well. And we were also able to see the restoration of cell spreading, lamellopodia formation, and localization of ezrin on the cytoskeleton. So previously I showed that these CF alveolar macrophages are releasing lactate dehydrogenase. So then we checked if this calpain also help in regulating the lactate dehydrogenase levels as well. So when we checked the lactate dehydrogenase level, we saw the significant reduction in their levels in CF knockout alveolar macrophages when they were exposed to LPS. So at the same time, we also assessed the functionality. When we assessed the phagocytosis of these CF alveolar macrophages in response to LPS and upon inhibition of calpain, we were able to see the improvement in their phagocytic efficiency. And in response to LPS, we see the less phagocytosis of these alveolar macrophages. And I would like to highlight that this is not only LPS dependent. Recently, Dr. Ma has shown that neutrophil elastase is also involved in failing the phagocytosis, and he did also inhibit the calpain and restored the phagocytosis. And further, we wanted to dig inside the specific form of cell death, what mechanism they are undergoing. So again, based on the literature survey, we found that when these alveolar macrophages are exposed to LPS, they are more prone to undergo pyroptosis. So pyroptosis is a programmed form of cell death, which happens in a canonical fashion and non-canonical way. In canonical ways, it involves the activation of inflammasome and the further downstream activation of caspase-1, which cleaves the gas dermin D and facilitates the pore formation via N-linked gas dermin D. And also it cleaves the pro-IL-1 beta and makes it as mature IL-1 beta and releases in the supernatant. And the non-canonical pathway involves the activation of caspase 11 by intracellular LPS, which again follows the similar downstream mechanism. So when we assess these markers in wild type and CF alveolar macrophages upon LPS exposure, we were able to see the increased expression of caspase 1 and cleaved gas dermin D and other inflammasome markers. So this suggests that these CF alveolar macrophages, when they were exposed to LPS, they were more prone to undergo pyroptosis. And then further, we asked if calpain is also involved in the promoting pyroptosis, because again, I showed the release of LDH levels. So LDH levels is another marker to say the cells are undergoing pyroptosis. So based on the literature survey, we found that calpain is indeed involved in promoting pyroptosis by releasing the sequestered pro-caspase-1 from the cytoskeleton. And also it was involved in directly cleaving the gas dermin D and promoting the pore formation. So further, we were curious to see what happens if we inhibit this calpain. Will it also regulate the pyroptosis? So we again use the same calpain inhibitor, calpeptin, to inhibit this calpain. And upon inhibition of the calpain, we were able to see the significant decrease in the expression of caspase-1, cleaved gas dermin D, and the other inflammasome markers, which were quite significant. So far in our study, we were able to develop a CF mouse model where we see the reduced number of alveolar macrophages when they were exposed to inflammatory stimuli. And using in vitro approaches in CF alveolar macrophage, we were able to show that upon LPS exposure, they have lesser ezrin levels, which affects their downstream signaling, promoting the cell death. And these cells are also susceptible, these CF alveolar macrophage are also more prone to undergo pyroptosis when they were exposed to LPS. And most importantly, we showed that the calpain is involved in down-regulating the ezrin as well as in promoting the pyroptosis in a different independent pathway fashion. And upon inhibition of calpain, we were able to restore the levels of ezrin, promote the cell survival abilities of this alveolar macrophage, and also improve the function. At the same time, we also reduced the pyroptosis in CF alveolar macrophages when they were exposed to LPS. So the next challenge is to see if this happens in the murine models as well. And also we would like to see if this inhibition of calpain in vivo setting of will it act as a therapeutic strategy in CF model of inflammation. I would like to thank Dr. Brucia for her continuous support and guidance, and I would like to thank the amazing lab members and my collaborators at Yale. And I would like to thank the Cystic Fibrosis Foundation and the postdoctoral fellowship. And also I would like to thank Katrina for these amazing illustrations. Thank you all.
See, we got a lot of questions. Good.
Look at that. Wow. That's what I'm talking about.
Very exciting talk, apparently.
Buckle up, Ravi. Buckle up. Let's go.
No, that was beautiful, Robbie. This is Katie Heiser from National Jewish Health. I'm curious which CFTR mouse or CF mouse you used. In your final picture, you had CFTR in the membrane just crossed out, and clearly, whether you actually have CFTR in the membrane and it's not functioning, or you don't have CFTR in the membrane at all, do you think that has anything to do with the mechanism? Does it matter what the mutation is?
So this model do not have any CFTR, so this is total knockout. So I think this mechanism happens independent of CFTR. At the same time, I don't know what exactly happened with the other mutation, but we have seen the similar decreased number of alveolar macrophage in G551D mice model, and we have seen the similar inflammatory effects, but we haven't explored the mechanistic aspects in those models. Maybe that will answer the question. Thank you.
Thank you. That's a great talk. I have two general questions. One is what is the underlying I guess biochemical mechanism linking the dysfunction of CFTR with the other effect in alveolar macrophage? Say they are prone to pyroptosis.
I'm sorry, can you?
What is the direct biochemical linkage between CFTR dysfunction and alveolar macrophage dysfunction?
So you are asking when there is no CFTR in alveolar macrophages, how does it enhance the dysfunctionality?
Right.
So I don't know exactly in the alveolar macrophage, but in macrophages, bone marrow derived macrophages, we have shown that loss of CFTR will impede the TLR4 pathway through ezrin, and that leads to their dysfunctionality, specifically phagocytosis. But in the alveolar macrophage, I'm sorry, I don't know it.
Yeah. In other words, if we restore CFTR function in alveolar macrophage, what could there be any benefit, therapeutic benefit?
Definitely, but since here we use the total knockout model, so the mechanism is happening independently of CFTR. So definitely restoration of CFTR it should be.
Okay. The second question is in the CF mouse models, are the numbers of alveolar macrophage comparable to those number of alveolar macrophage in wild type mouse? The number of alveolar macrophage comparable in wild type mice versus CF mice?
They were less in CF mice.
A little bit less.
Post LPS, but before LPS they were comparable.
Okay, before.
Before LPS they were all same, so there were no changes in the number. It happens only after LPS, and also it happens after 3 times of LPS. There has to be sequential exposure of LPS.
I see.
We did do 1-time LPS. We did not see any changes.
I see. Thank you.
Remember to say your name and where you come from so that we all know that you're part of our tribe and we can talk to you in the hallway later.
Thank you.
Hi, Irina Meralda from Emory. I may have missed this. I have 2 questions. Do you see the same effect with Ezrin loss in monocyte-derived macrophages, like the newcomers? And then also, do you think that actin dysfunction affects the alveolar macrophage's ability to patrol the area? And you think that that could be one of the ways they're dying, is just kind of falling apart as they're migrating through the tissue?
Definitely. As per the first question, yes, we have explored the role of ezrin in monocyte-derived macrophages. The mechanisms are quite different. When there is loss of ezrin in monocyte-derived macrophages, they were highly, especially the monocyte-derived macrophages were highly resilient to LPS. The effects were not like this. They didn't show the cell death and all. But we have seen the effects in the monocytes. So where they undergo apoptosis when there is no ezrin and when they are exposed to LPS. And also they play a major role in the transition phase when the monocytes go to the lung and they try to differentiate towards interstitial macrophages. During the transitory stage, the ezrin plays major role. So they reduces the proliferation effects as well as they promote them to undergo more cell death. So yeah. And I'm sorry, the second question. Definitely, since we see the defect in the cytoskeleton rearrangement, so we that is quite predominant. So they don't migrate properly. We didn't do the migration assay, honestly. So maybe if I had to be answerable to, like 100% sure, we have to perform that assay.
All right, let's get these last 2 quick questions and then we'll move on to our last speaker.
Thank you, Ravi. This is Tiro Vanziam from Emory and Georgia Tech. So I was wondering, you mentioned a little bit in passing towards the end, some experiments done with elastase as well. And my expectation, this is kind of the Pasteur Institute pathway that they discovered like maybe 10, 15 years ago, right? When elastase is being released, taken up by the macrophages, cleaves off calpain intracellularly, and then calpain is getting activated and does a lot of things afterwards. Is that the case? And if so, can you just use either a neutrophil influx inhibitor or, maybe we could use a GRIM inhibitor, I don't know, but also just simply a small molecule, inhibitor like civelestat and just get rid of the effect?
That's a great question. The thing about calpain is it's not just activated by one substrate. It has a multi-substrate components because calpain is required for phagocytotic efficiency. So they need to be activated at some level of degree. So the one that we saw is directly activated by calcium, and the calcium levels can be upregulated by other source as well, like from the other TLR pathways apart from the neutrophil elastase. So, I'm sorry.
The repeated LPS exposure.
Yes.
You see the effect primarily as a buildup of elastase. Is that what you see when you repeat the exposure?
We haven't checked the buildup of it actually, but we do see a increased number of neutrophils. Yeah, definitely. So, but as per the buildup, we haven't checked. So that.
Jonathan Zablok,
Hi, thank you.
KUMC. Actually, I had related questions about the calpain. Calpains have a very high KD for calcium, and it probably really matters which calcium channel you're really activating, right? Because it has to be some kind of a coupling to the calpain.
Yeah.
Do you know which channel you're looking at?
There is a nice study by Dr. Rob Terran. So where...
Who's that?
Sorry. They have shown that ORI1 is involved in upregulating the intracellular calcium levels, and they use this peptide inhibitor. If I'm not wrong, it's ELD 600.
6 or 7.
So with that inhibitor, so they were able to reduce the intracellular calcium levels and also downregulate the calpain activity. I'm not sure if they have looked at the inflammatory aspects as well, but yeah, that's another ideal target.
I'm not sure that ORI1 routinely activates calpain, but
yeah, so there are so many channels, so it can come from anywhere. So yeah.
All right.
Thank you, Robbie.
Thank you. Wow, we went from Rabin's Grimm's to Rob Terran's Ori, Emanuela's macrophages. I mean, this session has it all. So we're gonna load up one more presentation. So let me pull it up here. So our last speaker couldn't be here. It's Mimi Saavedra, Mimi Saavedra from the Colorado Adult CF program couldn't be here because of a family emergency, but she went to incredible lengths to make sure you didn't miss out on her contribution to the talk. So let's just play this and make sure that the volume is okay. And
good morning. I'd like to thank the moderators for inviting me to speak at this session, and I'd like to send my regrets that I couldn't be there with you in person today. I'm going to be talking about memory T cell metabolism, both in the pre-Trikafta and the post-Trikafta era. What I'd like to emphasize is that this is the work of Noel Raisavi, who has worked tirelessly over years' time in order to really try to understand what is happening in CF immunity directly from patient samples. And so I'm going to be showing all of her work to you here today. I have no relationships to disclose. So why talk about memory? And these x-rays all come from a patient who is taking Trikafta. And you can see here that she's suffering a pulmonary exacerbation. You can see the areas of infection. You can see them improve 6 months later. And then 6 months later, again, come back with an increased intensity and severity. And so the question is, what immunologic memory has developed so that someone can continue to suffer recurring infections with Pseudomonas aeruginosa without immunologic protection? What has happened to their adaptive immunity? When we talk about memory, we're not talking about the primary response to an antigen. We're talking about the secondary response, the tertiary response to that antigen, the quaternary response, so that after first exposure, you do get a memory cell in an effector population, but once you get re-exposure to that antigen, you get even a more robust and more efficient response, more memory cells, more effector cells, and these cells continuously recirculate from lymph node to tissues to blood, back to lymph node again, so that they're a very valuable source of information that you can directly take from the blood in order to understand how the host is responding to recurrent infections. One thing that's very important to discuss is that activation of T cells creates a huge metabolic demand. And that's shown here in this cartoon where you look at an activated T cell, which is making, this is mind-boggling, 800,000 proteins per minute in comparison to a naive cell, which is making 60,000 proteins per minute. There is a lot of nutrient uptake that needs to happen for this to occur. There is an order of 1 to 2 million ribosomes working necessarily in order to create 4 amino acids per second in comparison to 400,000 in a naive T cell making 0.8 amino acids per second. So huge metabolic demand. One of the most important metabolic substrates when we talk about T cell activation in CF are polyamines. You can see them here in green. They are byproducts of amino acids, L-arginine and L-ornithine. And there is there's catalyzation by the enzyme ornithine decarboxylase 1, which helps produce these polyamines. Polyamines are fundamentally essential in directing TH lineage commitment. So what is that? How does that relate back to CF? Well, polyamines have been associated with CF airway inflammation. Polyamines increase with CF exacerbations in the airway, and polyamines are increased in lung damage. So logically, our hypothesis was that pathways relating to polyamine metabolism in T cells would associate with T memory inflammation at the time of pulmonary exacerbations. Now, all this data was done before Trikafta when patients had a lot more pulmonary exacerbations. And so we took blood samples in patients who were adults with CF, both at the beginning of treatment of exacerbation, at the end of treatment. We isolated PBMCs. We enriched for CD4, CD45 RO positive memory cells, Ex vivo antibody labeled them immediately to get a snapshot of what was happening at that point in time. But we also wanted to tickle their memory and probe it. And so we in vitro stimulated them overnight with CD3, CD28 agonistic antibodies, then labeled them and then looked at them by flow as well. Let me talk about the study patients for a second. So there were 77 patients, and we divided them into a group that returned to baseline. So baseline FEV1, so 90% or greater of their best baseline FEV1 in that prior year, or patients who didn't return to baseline, so less than 90% of their best FEV1 for that year. And you can see here that pre and post treatment, the return to baseline FEV1, they had higher FEV1s. However, there were not significant differences in the use of steroids, in their viral positivity, in their Rosenfeld scores, in their CRP, or in their time to next exacerbation. This is our gating strategy. And so you can see from the single cells, the live cells, the CD3 T cells, CD4, the memory cells from that population, CD45RO, and within that population, you can see some IL-17A positive cells. So using that flow data, we created bar plots. And so on the y-axis, you can see percent expression by the CD4 memory cells. And then on the x-axis, division between the groups that returned to baseline FEV1 and the groups that did not in the pre and post antibiotic time points. And what you can see is that the groups that did not return to baseline FEV1 had higher IL-4, so more Th2, higher IL-17, so more Th17, and greater markers of T cell exhaustion, so greater PD-1. Since we had a lot of patient data, we were able to do multiple logistic regression analysis of predictors of return to baseline FEV1. And what we saw was that T cell lineage and T cell exhaustion were significantly associated with patients' inability to return their baseline FEV1. We then looked at ODC1 in these patients. And so using qPCR, we looked at their pre-treatment and post-treatment expression of ODC1 in the CD4 memory cells. They did not significantly decline after treatment, though they were significantly higher than the expression in healthy donors. When we subdivided those patients between return to baseline and no return to baseline, we saw that the patients who don't return to baseline had greater expression of ODC1 that was significant. So summary up to here is that ODC1 doesn't decline in CF CD4 memory T cells in patients getting PEX treatment. Even after treatment, it's higher in CF than it is in healthy donors. And there is higher expression in CD4 memory T cells in patients who don't return to baseline lung function. Well, it's already known that activated and proliferating CD4 memory T cells rely on ODC1 for biomass and growth. So maybe what it's saying is that just that there's more activation in these patients who are sicker and aren't getting back to their baseline FEV1. What is different is that having memory CD4 T cells activated in this way with these lineages appears to be figurative in CF infection outcomes. Let's talk about a second experiment, and this is looking at patients after ETI. So what happens to ODC after ETI? This is a completely different experiment in which we took patients before and after Trikafta and did single cell sequencing on their CD3 T cell populations and looked at expression of genes before and after treatment. And what we found when we looked at all these CD3 subsets is that ODC1 is down in CD4 memory and down in CD4 naive cells after treatment with ETI. What about hospitalizations? Well, in that population, as hospitalizations rise, ODC1 rises. So this is even after ETI. So let's talk about nutrients. And so you can imagine here that all this nutrient uptake in order to make more proteins is going to need a lot of metabolic energy. And under activation conditions, the currency for that ATP or that energy is going to come from glycolytic pathways versus during quiescent episodes or quiescent time periods, it's going to come from oxidative phosphorylation. Well, let's talk about bioenergetics and CF lymphocytes. And I'm going to show you here data from humans, data from mice, and data from cells, in which we're looking at bioenergetics in a CFTR specific fashion. So first, the humans. And so what you can see here is that this group of 4 CF patients versus this healthy donor, all of the CF patients who are taking Trikafta, none of them achieve the same amount of oxidative phosphorylation or mitochondrial respiration as you see in the healthy donor. None of them achieve the same rates of glycolysis as you see in the healthy donor. The patients are messy. So we then went to the CF mouse, and courtesy of Dr. Preston Bratcher, we were able to harvest splenocytes and look at, using seahorse, the production of ATP by those CD3 positive splenocytes in mice that had CFTR knocked out versus mice that did not. And you can see reduced ATP production in those CFTR knockout mice. Then we did CRISPR. And so we took healthy CD3 T cells. We, using CRISPR-Cas9, knocked down about 80% of CFTR in those cells. And what we saw was that in the CF conditions, there was still, again, reduced oxygen consumption rates, reduced oxidative phosphorylation. There was reduced glycolytic capacity in the CFTR knockout conditions and reduced glycolytic reserve. So overall, impaired bioenergetics. So in conclusion, remember, it takes energy to remember. So polyamine metabolism, we know it's fundamental to TH lineage commitment. And ODC1, as the sentinel gene in polyamine synthesis, has elevated expression in CF pulmonary exacerbations and in patients who don't return to baseline FEV1. CD4 memory lineage and CD4 exhaustion is associated with failure to return to baseline. ODC1 still rises with hospitalizations even after ETI. So I think the message I'd like to drive home is that you can talk about changes in certain nutrients. You can talk about changes in polyamine synthesis, but it's very important to take a step back and look at impaired bioenergetics overall. And so fundamentally, T cell metabolism is dampened in CF, and that may be a continuing target in improving CF infection outcomes. I'd like to thank everyone that was involved in producing this work, in particular Noelle, who has spent years doing this work, Katie and Sylvia, who collected the samples, Dr. Bratcher, who provided the mice, the sequencing core, Brian Vestal, who did the data analysis, all of our advisors, funding from the foundation, and of course our patients. Thank you.
I think we got 15 minutes left. If there are any more questions for our speakers, which I would like to thank one more time for their amazing talks. I think, go ahead.
Great job, guys. Thank you so much for sticking it out until the end, guys. Well done.